Hi, Shenshen. How are
Hey,
you?
John. I'm okay. So let me. Can you hear me fine?
Yes. Yeah, sounds okay.
Hear the construction in the background?
I hear no construction right now.
Okay. Yeah, I hear it. So yeah, it's been difficult. It's
Oh
like
no.
you are waking up every day on time.
Oh no. Are they doing something on like a building nearby or?
New dorms for students.
Oh, okay.
It's like it's been about over a year. It's synchronized with the virus.
A year is hard.
Yeah.
That's difficult.
Okay. But it's fine because I think if you can hear it, then I can use my earphone.
No, I can't hear anything in the background. I mean, maybe it's my computer, but it seems like everything is good. So
Okay. So maybe I should try my slide in case.
sure.
So let me try to share it. Is it? Let me try.
Yes, looks good.
It's working.
Oh wait, now it's showing the presenter view though.
Display settings. Would that work?
There we go. Yes, now it looks good.
Working.
Yeah,
Okay. Okay. So it's moving. Right.
perfect.
Yeah, it's good. I checked the time because we have it at 4.
Right,
I assume.
right. Yes, and of course, one of the things that has not changed between now in the Zoom era and previously when everything was live is that it always takes time for people to filter in. So we usually
Definitely.
after a few minutes. So we'll wait for people to come on by. So are you all? Gosh, are you all in the same schedule as us? Is this the last week of the quarter for you all as well?
Yes, so it's our last lecture today, actually. My last. Yeah, the Zoom teaching is unfortunately also ideal for the students and maybe for us.
Yeah,
See anyone.
not the best for anybody. I think that's true. But
In the fall, it seems promising that we can go back partially.
yeah, it seems like it. It looks like over here in Riverside, most things are going to be back in person in the fall. At least that's the plan right now. Barring some kind of terrible things happening in the summer, but seems good. Hopefully things are optimistic over there as well.
Yeah, we have some very big classes, so it's not yet like.
Yeah, so some of those might be either online or hybrid or something like that. We're hoping that will also resolve soon. Oh, there's so I'm seeing a message in the chat. Is there an issue with the link to the colloquium? Actually, I don't know if I can I don't know if this is a problem that I can fix.
Well, I don't know about the rest of us, but for some of us, the new colloquia have not been announced.
Oh no.
Yeah.
All right. Well, hopefully, I got a message earlier today.
Well, apparently that message has not reached graduate students, at least not reached graduate students in my class. I've just learned about that in my office hour. So.
Oh no. Okay. All right. So we'll have to talk about we'll have a discussion about the email list. That's unfortunate. I don't know if there's a way to quickly disseminate because I don't have the list. And then it's already the time is now. But hopefully we'll have a good group of people who can join. We give maybe a couple more minutes for everyone to get here.
Yeah, maybe it's my fault. I sent you my abstract like last week.
Oh, no, no. I'm certainly not. This is sounds like an email list issue. Email list and perhaps, you know, general end of quarter fatigue. But we're making it through. All right, so maybe Shenshen, if you're okay, I'll go ahead and start the introduction. Sounds all right. Okay, great. So today we're really pleased to welcome Shenshen Wang for the final colloquium of the quarter this year. So she got her bachelor's degree from Nanjing University before moving on to a master's degree from the Hong Kong University of Science and Technology. She later got her PhD in physics from UCSD before a postdoc at MIT. And now she is an assistant professor of physics at UCLA nearby. And her work uses ideas and techniques from statistical physics to study primarily the immune system. She also has interests in problems like in active matter. And she's given talks all over the world in this area and has received multiple different awards for this work, including things like a Career Development Award from UCLA. And we're really happy to have her here for our final talk. And I'm excited to hear her research. So let me turn it over to Shenshen.
Thanks very much, John, for this invitation. And I would have loved to meet you all in person, especially we're only less than 2 hours apart. But I'm happy to meet you here toward the end of the quarter. So I really appreciate that you make the time to join me. So please ask questions during the talk. So the point is really not to get through everything, but to for you to maybe have some new thoughts about a topic maybe you don't normally think about, but maybe during this year is more often on our mind. So I really want to tell you about some ways we think about the immune system, especially the adaptive immune system, which can actually evolve and adapt to a complex environment, how we think about it from a more physicist perspective. So as John mentioned, so I have been interested in two particular broad range, I would say there are broad families of systems, even though they're under a particular name. So on one side, I worked on thinking about systems that are made of many interacting components that collectively make up the interior of the cell. So it's now under the name active matter. So it's really talking about systems composed of self-driven units that consume, in this case, chemical energy and turn it into mechanical work. So it's an internally non-equilibrium system in this sense. It's called cytoskeleton. So it's a biopolymer network that supports most of the cell's function, including how they move in space, how they interact with each other and their mechanical environment, and including like when we contract our muscle cell, all these little machines called molecular motors So can you see my cursor? Like here, these little engines that acts inside this biopolymer network that allows all this macroscopic function to happen. So their coordination by doing local things is a very important and interesting puzzle that people are still pondering through. So on the other hand, like John said, we both worked on immune system for some years. For me, an entry point to the field, or something I really draw me to this complex world, is something called affinity maturation that I will tell you a lot more about. It's really the process that's happening inside us, which makes antibodies and therefore make the vaccine hopefully work. So it's a Darwinian evolution process that happens really at a striking speed, because we need to make useful antibodies when we get flu inside us. So over weeks, we will make pretty good antibodies that can recognize and fend off the virus if we see it again. Okay, so the immune memory will be generated. So in this sense, it's an externally driven population of cells that evolve at very rapid speed. So it's different from most of the evolution process we thought about, which really involves timescales like tens of years over a lifetime of mammal, or even over millions of years. If you think about the HIV virus, the more ancient virus. So it really involves many time and land scales that allow us to generate this internal evolutionary process. But the drive of these two classes of systems are pretty different. So one is internal, the other is external. But they share in common that these non-equilibrium forces can generate or yield very different collective states that near equilibrium system wouldn't be able to find. Okay, so those will be the general theme I will try to convey through more specific examples. So I think over the past year, we're fully aware of the fact that we're sharing this universe with many other species, in particular, these fast evolving species like viruses. So here I'm showing some pretty examples. So is there some questions? Heard something. So here are some pretty examples of the phylogeny of different viruses. So that means it's pretty much like the descendants, the family tree of, for example, HIV. So you see this radiant pattern. Each branch is a specific, distinct viral strain. And you can see the morphology and complexity vary a lot across this viral world. So HIV and HCV, which are infecting us and evolving within us, they have this shared radiant morphology. But on the other hand, if we look at influenza, it's a very linear kind of pattern. Sometimes it branches, but mostly it has this very distinct morphology compared to HIV. So it's like a more lower dimensional path in this state space. So in this sense, you would wonder what leads to this dramatically different complexity and morphology of the viral trees. So the truth is, part of the reason is it's also driven by their host. So in this case, HIV and HCV, they evolve inside a person, for example. So it's within that single host. It's driven by a particular immune system of that person. On the other hand, the flu is just circling around across the globe. So it's driven by, in some sense, a collective immune system of many of the people, animals that are transferring this virus very efficiently between them. So it's evolving under a very different pressure and time and a land scale, as you can imagine. Right? So it's like over 40 years. So I will tell you more from the immune side, but we keep in mind what's driving the immune system is this pathogen world that the immune system has to face and try to solve the problems that are presented with. The difficulty about the task the immune system needs to solve is not foreseeable, right? So they don't know what's coming next. The best thing they can try is to learn about some statistical structure of the pathogens they encounter in the past and try to predict what may come next. So it's a pretty difficult task because each individual person, it's impossible that we can individually sample the statistics sufficiently so that the prediction can be made in an informed way. So I will tell you primarily two levels of examples. The first is really the very first step of any immune response, because as you can imagine, the first step has to happen in a sense physically. So the immune cells actually, they are equipped with surface receptors, and they basically touch their antigenic targets physically. So here, I hope this can show, is showing one branch of the immune cells, the adaptive immune system.
The green cell you see here is a killer T cell in the immune system, which is attacking the cancerous vertebrate cell. You can tell when the killer cell is very basic.
So the point here is you can see first this T cell, which is called killer cell because it's not directly destroying the pathogen itself, but it tries to remove the infected cell itself, which is infected by the virus, and present some little tag on the surface of the infected cell. So, but you see it's a very tactile sense, right? It's spreading this protrusions that try to wrap around the cell surface to detect the antigen presented on the surface. So antigen is a little part of the pathogen that can be recognized by the immune receptor. So on the other hand, B cell is the other branch. So there are two arms, B cell and T cells, which make up our adaptive immune system. So the B cell are using similar physical ways to probe their target cell. So you can see this fluorescent labeled antigen presenting cell, which is in blue, and the B cells, which are much smaller in red. And these overlapping region in yellow is where this so-called immune synapse will form, which is a dynamic motif that mediate the interaction, communication between the cells. So this communication actually happens through multiple pathways. It can, first of all, is physical binding interaction, and that will then lead to biophysical signaling inside the cell. There's some network that will respond and try to mediate downstream process of the cell. And also, more strikingly, there will be material flux, meaning if you look at this fluorescence image, this red, this yellow is indicating the antigens. You can see the B cell is physically grab a little part of the antigen and then internalize it. So this is the B cell surface. And that will then lead to these downstream processes. But the first step is a physical contact and subsequent internalization of the antigen recognized by the B cell receptor. So I'll try to convince you this is a first physical step. And T cells and B cells, the reason we have two arms, of course, is because they have distinct and cooperative functions. So on the one hand, the T cells, they are once they are released into our bloodstream, they're already ready, they're already trained to recognize a huge diversity of peptides presented on the infected cell. And they will need to make decisions like life-death decision, whether that cell should be killed or kept intact. So this is like a cartoon of the process that if the T cell receptor recognize the antigen, then that will mediate some pretty complex process that try to basically kill the cell into that state. So that's a very binary decision. On the other hand, the B cells, they're not pre-trained. The reason is this branch is able to adapt to the new invaders they see in real time, meaning on the fly. So they undergo this Darwinian process called affinity maturation. So let me tell you a little bit about it. Upon activation, the naive cells, which haven't experienced any pathogens or of this type, the new invaders, if they are able to recognize the antigen, they get activated. And a bunch of these activated cells will coalesce into a population, forming a class of cells locally inside our tissue, for example, lymph node. Then they will replicate and make copy errors at a very high rate. So the error rate, so-called mutation rate, is about 1 million times higher than that of the normal tissue cells in our body. Okay. And this error-making process will allow them to generate a huge diversity in a short time. And this newly altered form versions of the receptor will then meet the antigens that are transported and presented on the surface of this presenting cell. And if they are successful in recognizing and internalizing those antigens, as we saw in the movie, then they will be able to compete for another type of signal. So that involves another kind of T cell that coordinates immune response. This helper T cell will decide which T cell will get the signal they need to survive. So the winner, usually the one which internalizes more antigen, will have multiple possible outcomes. So either they go back to go through further rounds of this replication, mutation, and selection process, or they can leave this structure or terminal center and become either memory cell or plasma cell. So the memory cell is what's monitoring the encounter of similar pathogens in the future, so that next time our body sees it, the response will be much faster and stronger than the first time the naive cell sees it. And the plasma cells, they are of different lifetimes, and the long-lived ones even enter our bone marrow and is stored as a longer-term memory in the sense. Once the body sees the antigen, then the plasma cell can secrete antibodies to remove the antigen. You can call neutralizing them. So the end outcome of this cyclic Darwinian selection process is first, we can generate higher and higher affinity antibodies that are better at recognizing the invaders. Second, the immune memory is generated. So in the future, we're protected. So that's what vaccine does. It elicits the memory so that when we see the real virus, where we will experience much milder symptoms. Okay? So these are the major picture of these two branches. And this physical contact between the immune cell and the antigen presenting cell, curiously, exhibits very different spatial pattern depending on the stage of development of the cell. So on the left are the naive or memory cells. So they form this single large cluster that is concentrated in the center of the contact area. So we are looking at the cross-section between two cells. For the maturing cells that are undergoing this evolution, this training process, they will form many segregated small clusters of antigen in the contact. So it's not clear what process generate this very distinct architecture of the contact pattern, and second, what's the function of it? Because the cell wouldn't bother using different patterns if it's not functionally meaningful. So for T cell, this has been observed. It's very similar to the contact or the synapse formed by T cell and their target cell. But this very segregated pattern, which can maintain this steady state before the B cell go to the next developmental stage, is something curious and not well understood. So a simple thing we can do as a physicist is to write down Hamiltonian that describe the major components of this process. Right? So we're thinking about what's the total energy that's involved in this contact region. Two components mainly. One is accounting for the elastic property of the membrane itself, where the receptors are embedded. So you can have bending and tension that accounts for the morphology of the surface. So it's costly to bend it because you are stretching locally elastic material. And these are like non-specific energetic term, but the specific interaction of multiple species involving the B cell receptor and the antigen, which are much shorter than some longer molecules, which are adhesion molecules that keep the cell in contact. And also there are some repulsive agents that prohibit the cells from sticking to each other. Okay, so if we account for this diversity of interacting agents, and write down interaction terms for each of this. And it's important that there's a diversity of height between the cells, between the molecules, so that you can see there's a cost to bend it when the bonds form between the short and the long molecules. So if we only account for this, it's just interaction like we usually expect. Due to this bending cost, the system will tend to form a single large cluster over a very long time because it's pretty much a nucleation process and drive the system toward a complete phase separation, meaning you have two completely segregated phase regions of, in this case, the receptor antigen binding region and the rest. In this case, the adhesive molecules will occupy the rest, because they want to minimize the surface interface between these two regions to reduce energy. But that won't be able to explain this multifocal pattern I showed you for the evolving cell. So here are some pretty recent experiments that try to probe what's happening during this contact and then extraction process. So they use this DNA-based tension probes, meaning, so this is a DNA hairpin. You can see they form pairs because they are interacting pairs, right? Bases. And then if at the beginning there's no force applied, so on the top there's antigen, at the bottom there's some glass substrate. So if there's no force from top, then this hairpin will be intact in its native conformation, so that there's a dark quencher that will suppress the fluorescence. So nothing interesting, you wouldn't see anything like a dark image. But once there's force or anything that disturb the system, pulling the antigen away from its natural configuration, then the hairpin unbreaks. It just unfolds so that the dark quencher is removed from this fluorescent label. So you will see this thing lights up, so as we saw here. So this is a very sensitive probe to measure molecular forces as small as several piconewton. So what we're seeing here is comparing naive and the maturing cell. You can see this fluorescence over time. That one stays green, meaning you only see this one. But this, you see the flashes of the red dots, which indicates the breaking of the hairpin and therefore force application in this vertical direction. So you have normal forces. So B cells are applying locally normal forces to pull on this receptor antigen bound and probe the interaction strengths in that individual molecular interaction. So we have to account for this force in some way and see what's the consequence. So in this sense, we're dealing with no longer an equilibrium membrane with only bending and stretching. We need to account for this mechanical active process due to the normal force. So the phenomenology that we know of basically are two. One is these clusters of receptors and antigen aggregates will grow and emerge before they eventually become arrested, meaning they stay at a certain size without further expansion. And subsequently, we know this clustering will instruct force application. So there's we know this phenomenologically, but physically we can think of mechanical energy due to these pulling forces acting on the bounds in this form. So only when the number of bounds in a cluster exceeds certain threshold size, then the pulling starts. So that's accounting for this delay between cluster formation and the force application. And third L is the induced membrane displacement. So roughly the picture is like this. The cluster grow and then pooling happens. But what's the outcome of the pooling is it's like a recycling because the force will limit the growth of this cluster in the lateral direction because some bounds will be broken due to the force. And at the same time, there will be a supply from the free antigen that will join these existing clusters or form a new one. So you can see there's a dynamic balance between formation of new clusters and breaking of existing ones. And turns out, once we consider this force application, you see is the red is the antigen that are bound by BCR, B-cell receptor, and the gray are the free ones. So that's a free reservoir. So you can see, first of all, the red clusters maintain pretty much similar size. But at the same time, there's constant material flow between the clusters. And here, when you see a dense free antigen cluster, that means it's like a remnant of a just broken red cluster. So it's a highly dynamic equilibrium. And if you look at distribution of cluster size, there's a distinct characteristic size, like we saw in experiments. If you look at 3D morphology, then there are these protruding structures, like people saw in the immune cell antigen presenting cell contact. And since this force-dependent process is able to capture the basic phenomenology, and if we put this on a kind of a diagram, if we plot the number of patches, meaning the red patches that indicate the bound BCR antigen complexes as a function of the binding affinity. If there's no force, you have this complete phase separation. So higher affinity means increasing, but then saturating bound region. But as force increases, you can see this coarsening transition will be delayed or pushed toward higher and higher affinity. The reason is it costs more energy to arrest this phase separation. So in this plateau region, that's where you have this dynamic, maintained multifocal pattern, like in the evolving cell. But if affinity increase further, you can overcome the force, then you will see this percolating clusters. So this force in the normal direction is able to modulate this lateral organization of receptors that are mobile in the surface. As a result, can tune the degree of phase separation. But what's this useful for? Because it costs energy to the cell. So one indication we get is if you look at the affinity range that can be distinguished, meaning the amount of bound complexes depend on affinity, we will see a stronger force application or energy input will lead to a wider range of affinity that are distinguishable to the B cell. At the same time, as you can imagine, the thing will be sped up because of the force application. Okay, kinetics gets faster. So this will be functionally meaningful because eventually the B cell need to evolve, meaning they need to rank themselves based on how good their receptors are. So this provides a potential explanation for why the cell actively modulate the force application to probe their target. But this process, like I said, is a very physical process, right? At the very beginning of the recognition. How that later influence the evolutionary efficiency of the cell as a collective, the population of cells. We have to think about how evolution depends on the physical interaction. So this is just to show you a more global picture of, we can have about tens of these local B cell receptor clusters that go through this maturation cycles inside a single lymph node. And we have hundreds of lymph nodes all across our body. So it's really a spatial temporally modulated process, and there can be cell migration that connect these populations. So it's a complex process, but some aspects are understandable from a physics perspective. So on this larger scale, imagine surface mechanics could be a useful framework, and so that's what I had been worked on to understand. But today I want to tell you about this antibody evolution in a broader context, in the sense that if you think about what the immune system has to have to solve, it faces this diversity and the evolving world of pathogens. Then for each pathogen, it has to decide what kind of receptor is useful in long run, right? Because most of the time, our antibodies are specific for a particular virus, like the influenza. But the reason we need the updated vaccine every year is because the virus is changing itself, is making a living against our immune detection, right? So in the long run, if we can generate something, some receptor that can recognize a diverse range of variants, different versions, mutated versions of the same virus, then that could be much more desirable. That's more protective in the longer run. But it's hard to do that for the immune system. The reason is, how do you select something good for the future? Okay? So this is a question about whether when a living system is facing a changing environment, whether it should specialize to some particular environmental condition, or it should use a more generalizable solution that can be good enough for many conditions, but in each particular one, it can be less fit or less of lower performance than the specialist. So this question has been thought about in many contexts, including microbes, because people found they can switch types between different so-called phenotypes through this kind of schematic way. But as the environment changes, you can think of the state of the system is trying to align with the environment. So as long as you have a subpopulation that's fit for each of this, eventually you can have a mixture of specialists that can deal with the environments you usually encounter. But this relies on the assumption that the system can sample sufficiently about their environment. And also these environments usually are unrelated. So this is a simple mathematical description of this process. This population vector will evolve in time according to a reproduction term, depending on how well the phenotype matches the environment. And also there can be switch between different phenotypes, like the transition matrix. So usually the transition matrix is written in a more empirical way. You can assume different structure of it. But here, the question I'm trying to ask is how is this empirical matrix can be emerging from more microscopic process, meaning how does evolution give rise to such a transition matrix? What determines the transition probability between a specialist versus generalist solution? And how does environmental dynamics affect their relative probability? So just some example of who are the generalists I'm talking about. So like for the B cell realm, there's a special class of antibodies called broadly neutralizing antibodies. And you may heard there are cross-reactive COVID antibodies. So that means that each single antibody can recognize a broad array of diverse variants of the virus. So even if the virus modified itself over time, the antibody is still able to catch it. So here is a dendrogram showing for HIV, when a particular person is tracked over time, you can see these colored branches indicate the antibody is now able to cover that particular strain if it turns colorful. So over a long period of more than 5 years, this individual is able to go from recognizing nothing to evolving an antibody that can recognize almost all the circulating strain. But this is very rare. Most of the people, they wouldn't be able to evolve antibodies like this, so the virus can easily escape the specialist antibodies we constantly make, but it's falling behind the virus. On the other hand, we heard of drug resistance. It is a big problem, like both in cancer cell or in bacteria. They have no trouble developing cross resistance to many different drugs. In this case, it's some bacteria isolated from soil, average numbers like 7 or 8, and antibiotics each bacterium will be resistant to. So then the question is, how come generalists can evolve very rarely and slowly in one case, but very abundant and quickly emerging in the other case? So what determines their evolutionary tendency? So this involves a mapping from the sequence, the DNA, RNA sequence that encode bacteria or the virus or the antibody, to their relevant function. So function can be defined in different contexts. In case of antibody, is how well they bind to their antigenic targets. So in recent year, there's this very quickly advancing technology called deep mutational scanning. So it's really trying to map out this relation from the sequence to their corresponding functional score using this kind of morphology metaphor of a landscape. So that means you're looking at a local neighborhood near a particular reference sequence, and you mutate in the lab to all the neighbors one mutation neighbors of a sequence. And this combinatorially complete local region around the reference will tell us how rugged it is, and in turn determine how difficult it is to evolve to a local peak, meaning higher fitness solution, starting from the sequence your experiment is set up for. So this kind of landscape has been mapped in many different systems, including enzyme, antibodies, protein binding, and RNA viruses, and their growth or infectivity. One thing about antibody antigen binding, so there is very hard to do in practice because this is combinatorial. So as the system increases, meaning the length of the sequence increases, then the size of this state space is exponentially exploding. But even within a tiny neighborhood, like up to 10 amino acids involved, people already observe something called epistasis. Basically, it's interaction between mutational effects. So what this tells us, individually deleterious mutations, meaning decreasing the functional score, can be jointly beneficial. In other words, this landscape can be pretty rugged. If you can cross a fitness valley, you can get to maybe a higher peak than you were originally at. And this study shows this epistatic effect contributes significantly to antigen-antibody binding. So it's proving that this ruggedness in a high-dimensional space seems to be a reasonable way to think about a system. So the question I was trying to answer is, if we have this rugged landscape, but for a particular antigen you have a particular landscape, what if the virus evolves or the immune system is seeing different landscapes over time? Would that lead to different kinds of solutions than if the system is evolving in a static way? So this is a simplistic picture. So anything I can draw in low dimension is pretty wrong, but this is one way to help us think about a system visibly. So if I start with a rugged landscape with multiple peaks separated by valleys, then you can see if I start at the red star, the red peak, then I switch to a different environment, meaning a different kinds of landscape topography, then the red star will fall on slope. So that means it has a chance to climb the hill nearby to the neighboring peak. So it will be mobilized by this switching of environment. And if I switch back to the original environment, either it can be trapped and return to the original peak, if that's the only better solution it can find when it switch back. But on the other hand, it's possible that it can find a different route to another peak. So you can see the difference between the red and the blue is if I further do this alternation between the landscape, the red and the gray in this local neighborhood will just be oscillating. But for the other one, it will find a path to find a new peak, which is stable across the environment. So you can see after we do this cycling between the environments, some of the solutions will be forming like a limit cycle, and other pathways will lead to a fixed point, meaning if we further do the cycling, it wouldn't perturb this subset of solutions that much. But the other one will constantly be driven away from where it is at the moment. So in this sense, we can classify this local optima, meaning possible solutions to the binding problem, into specialist and the generalist subsets. And their key difference is how much they respond to the environmental changes. So generalist has a much smaller fitness variance upon this environmental switching. In that sense, they are living in a cooler space in this high dimensional state space. But how do we, if we want to evolve this generalist in the context of antibodies, how can we enhance this kind of asymmetric transition flux between specialists and the generalists? Because we don't want the other way around. So the answer I will tell you very quickly are from two aspects. One is topography, meaning how the landscape surface guides the evolution trajectory. The other is dynamics, meaning the time scale of the environmental changes. So people can construct rugged landscapes using analogous formula to people construct spin glass, but the shared property is you have interaction of different orders between the sites. The spin, in this case, the spin flip represents the mutations. So what's surprising here is if we, so remember, have these alternating landscapes, if I change their similarity, so NP along the x-axis, we increase the similarity between the rugged landscapes. And toward going upward, I increase the ruggedness, which in this case means how many neighbors are coupled, represented by K. What's surprising is not that the fraction of shared local optima is increasing when we increase similarity, right? That's expected. But what's more surprising is as the ruggedness increase, the chance of sharing local optima will be higher. So this actually reflecting that the morphology of the topography of the landscape depends on this coupling factor, the degree of coupling, the range of coupling among the mutation effects. High ruggedness not only increase the number of local peaks, it also affects how they are organized. In this case, they are more evenly distributed and therefore increasing the chance that two peaks are within close proximity. The other important consequence of this switching is compared to a static environment where we can define basin of attractor of each peak, local peak. When we have two environments that are related but different, so you can have a different set of basin of attractions. But when you switch between them, you can see if the peak in one environment falls in the basin of the other, then the system will be pushed away from the original peak to climb to a neighboring peak in the alternative environment. So eventually, if we do this cycling, the system will be driven toward the peak that are close by, meaning they are basically shared across environments. So you can see dynamically it's possible to drive a biased flux towards this generalist. As a consequence, if we can place the system on a phase diagram, like how related the landscapes are and how rugged they are, then we can have two kind of phases. One is this limicycle dominant one, the other is this fixed point dominant one. So what this tells us is it's possible to avoid this multi-drug resistance by pushing the system into phase one, or trap them in the very rugged extreme. But in between, there's a high chance we evolve fit generalists. The reason is you can expand the size of the basin by connecting them dynamically, like I showed you earlier. The discrete, disjoint basins become connected due to the dynamic cycling. And this is the chance to evolve generalist antibodies. You can locate the system in this region. And so what I told you is this global statistical structure of the landscape can give a chance to drive system to places where they don't normally go to. And if we know where we want the antibodies to evolve, we want a particular sequence, then we can encode them using a construction similar to what Hopview did in the neural network. But the idea is you can encode the target, the antigenic target, into this antibody binding space. So different peaks represent the target epitopes. And different antigens can share some epitope, but differ in others. So we want to drive the system to this shared cross-section of the islands, which represent broad antibodies. The conceptual picture is simple, which is the environmental timescale is key to keep the system asymmetric in their flux between specialists and generalists. Because if we switch very quickly, you can imagine the system have no chance to explore further than their immediate neighborhood. So they will be trapped close to where they start from. But if we allow more time for the system to divert from where they were, then there's a larger leakage from the specialist to the generalist. Because as I said, specialist regions are hotter and generalists are cooler, so the system will collect to this more stable region that are insensitive to environmental fluctuations. And the revert flow is much smaller. So there will be an intermediate timescale that both allow evolving enough time for the system to find the generalist peak and also maintaining it by switching not too slowly, because too slow switch will allow the system to escape from the generalist by crossing the valleys. So there will be an intermediate timescale range that allow this balance. And we did this with HIV data from published work. So we can see a partial tree of HIV antibodies evolved in a person. So we can group things into specialist antibodies that are unique to a particular viral strain. And one of them is shared between the two strains. And when we do cycling of the antigen, we see indeed it can evolve broad antibodies. But in contrast, if we present them simultaneously, in this case, if we cycle between them very quickly, then there's no success. And also, interestingly, if the specialists are less correlated, it can drive a larger variance of this cycling induced population spread. So that will help with finding the generalist solution. If we shuffle the antigen antibody assignment so that we artificially increase the correlation between the specialists, then the success is reduced. So both the similarity between the environment and the timescale of cycling are key to evolve. And you can think of a larger space of dynamic strategy, including what we tried called chirped cycling. So you increase the frequency of cycling over time so that you allow more time for the population to find a solution so they can survive this cycling. And then you speed up, because then you can more effectively localize them to the solutions they found. So this way, things get more efficient and more robust. And you don't need to fine-tune the timescale of cycling. So in a nutshell, what I told you is at very fast and slow switching, the system can be described by an effectively static landscape. But in between, the system is really partially relaxing. So they're not allowed to reach their steady state. So that's the truly non-equilibrium regime where there's opportunity to drive the system to underexplored region that they normally go to under effectively static environments. So I think I should wrap up. So this I can say in 1 minute, if the environment is itself evolving, like the virus that can evolve quickly inside their host, HIV, then an interesting observation is you can have very different viral outcomes. For HIV, there's no success in any person ever in controlling the virus. So there's always this red queen like chasing dynamics and characteristic oscillation in the viral abundance inside a person. But in contrast, HCV, which is very similar in diversity in their strategy to escape immune recognition, there are people, about 20 to 30% of people, they can develop very different trajectories of the virus. Some are able to spontaneously eliminate the virus, either directly or after some viral rebound, eventually can control them. So we study how can we think about this dramatically different outcomes by including the feedback from the viral evolution to the B cell maturation. Okay, and that allows us to describe the system in a recognition space with dimensions where system can evolve with different speed to reflect the heterogeneous cost of mutation, because some sites are easier to mutate for the virus, it's less costly to its fitness, and then they are moving faster along those directions. But some sites are essential for their function, so in those directions they move slowly. So this kind of description allows us to predict what are the important factors that determine the outcome. So we find the conservation of the virus and their initial diversity, meaning when our immune response starts, basically determine the outcome. So you can see these red queen type oscillations, or a very rapid bottleneck and then rebound, or a monotonic clearance, depending on the combination of these key parameters. But the key is the different response modes. Like some are faster, which are specific, some are slower, the broad ones, are coupled through the antigen dynamics, like a shared resource between the cells. So to wrap up, I hope I make some impression that physicists can think about the immune system and contribute to this thinking in a different way than biologists usually do. So 2 examples I showed you is the very first step of immune recognition is very physical. So this physical extraction of antigenic information sets what all these following steps can process upon. So that's really like a limit of information at the very beginning set physically. And then I showed you this internal motor-dependent cytoskeleton-driven process that allow force application is essential to organize the interface between immune cell and their external world, and therefore keep the immune system out of equilibrium to probe their antigenic target more efficiently. And the external driving force from this co-evolving pathogen universe is also acting to keep our immune system off equilibrium. So this generalist solution, as I showed you, is one consequence of this environmental dynamics. And the time scale relative to the intrinsic dynamics of the immune system is what determines where evolution will drive the system inside the state space. And finally, this co-evolving environment can drive very divergent outcome due to the feedback between the system and the responding or adaptive environment it's facing. These are the people who did the work. I'm highlighting the students, graduate students who are doing the synapse work with a postdoc, former postdoc, and Jiming is the one who's responsible for the coevolution part. I didn't have much time for, and we also collaborate with Arvin's group on this generalist evolution project. Some other papers we work with Lei Yan at BioHub at the time. And Lei was at UCLA, but now he's a group leader back in China. And we worked on this rugged landscape switching project. So yeah, so I think that's, if you're interested, there are some work we published in recent years on the topics I tell you about. Okay, so yeah, sorry, a little bit over time, and I'm happy to answer any question you have.
All right, thank you very much, Shenshen, for a really interesting talk. It's great to go, all the way from the really physical to modeling these sort of more abstract processes in the immune system. So let's see if anyone has questions. You're free to, you can either raise your hand in the Zoom, or you could type in the chat, or you can just ask a question. And maybe while people are doing that, maybe I can start with a question over here. So one of the things that I was wondering about when you were describing this, organization of the immunological synapse and how it's changes from this sort of state where everything is clustered together to having these little micro clusters that go around. Is there some possibility? Do you think that this might be something that's, that might be controllable externally for, say through, if you're to give someone a drug or something like that, to actually help to kind of control these processes?
It's a very interesting question. I think from the more physical perspective, this fourth generation is key to control this process. So I think the new point we try to make is cytoskeleton force can be playing a very active role rather than a supportive role as people originally thought about. So the active role, by that I mean, they both control the formation of the cluster and the later extraction. So if there's a way to control the fourth generation, for example, using some drug to modulate the pathway of fourth generation, that could be one harder possibility because it's internal, it's involving many other processes. The force can be useful for other processes. But externally, I think it's easier and more promising by modulating the physical environment. Because I didn't mention for example, the stiffness of the surface, how dense the antigens are presented are all important because of this very interesting, it's like a tug of war configuration, receptor and presenting surface.
So maybe in terms of how I guess even if you were to present those in a non-native context, so not directly on the immune cells, how you what kind of structure you use to do that presenting could have a really big effect on what the outcome is.
Yeah, people are trying that, but of course you would say, oh, but inside the person, how would you want to perturb that? That's always a hard question. But at least we can understand better how this probing process works by in vitro constructing different presenting configuration and see how much effect that can cause and how much, because the cell, there's one interesting thing. The innate system can send out some signal to control like stiffness of the presenting cell. Like under inflammation, there's some experiments showing the presenting cell will get stiffer. That seems to be an interesting modulation or communication between them. So the adaptive system will be more stringent in their antigen discrimination due to the stiffness response. So
that's really interesting.
Yeah, quite physical in the sense that potentially the immune system is already using it to modulate the response.
Yeah, and maybe so. Let me pause for a moment here. I don't want to run over people. Let's see if there are any other questions. Okay, if there's nothing immediately, maybe I can follow up on that briefly. One thing I've heard some things about recently is, people when they're studying things like cancer cells, for example, they often find that they have that they actually have different mechanical properties than like normal healthy cells. So it would be interesting. I wonder to what extent people have explored how that just those mechanical differences might actually, for example, help cancer to escape from being controlled by immune cells.
That's right. I think that's very interesting because there's indeed difference between cancer cell and healthy cell in terms of their stiffness, their composition of the membrane, I think. So I would imagine then that would directly affect how T cells are able to recognize the neoantigen maybe, and whether they apply force to try to distinguish the cells when they meet a cancer cell than a healthy cell. Yeah, I don't know the answer, but I think that's super interesting. And the cancer cell, they migrate. So that puts some constraint on their membrane organization as well.
Sure. Yeah, of course. There's so many things to
Yeah.
that could happen, right? But it's very interesting.
Yeah, but I think it's not yet thought that much about how this may have consequence on the immune
Mhm.
regulation.
Yeah, absolutely. All right. Well,
Great.
if there are no other questions, then let me go ahead again and thank Shenshen very much for a really interesting talk. It's great to have you here to close out the quarter, and hopefully there'll be times to interact, in real life in the near future as well. But yeah, thanks very much again, and thanks everyone for coming. And look forward to catching everything up again in the fall.
Thank you very much, John, for having me. And thanks everyone for attending. I know it's hard to make a time at the end of the quarter. So thank you very much. Hope to meet you at some point.
Yeah, absolutely. All right. Thanks. Take care, everyone.